Welcome to BioMars.
Hello everyone, today I'm gonna talk about meiosis. So basically meiosis is produce the next generation. Basically it should be germ or egg cells. And the mechanism is 2N form 4N. It's not like 2N form 4N. It's 4 small n, 4 single n. So the cell is 2N and each small cells are N. And the process is so first is a twin cell, and DNA replication happens, so each one of them have the sister chromatid. So sister chromatids are identical cell, like they are identical, and they are also identical. Also the yellow one, and the crossover happened. I will talk about crossover in more detail in the later. And for this stage is meiosis one. So crossover happened in the meiosis one, and then the anaphase one is each cell segregate. Also, in the metaphase one, on the mitosis is like this one. There are sister chromatids in the between in a line, but in the meiosis they are separate. Look, there are two on the left and there are two on the right. And then the anaphase is separate the four sister chromatids to two parts. And in this one part, there is a blue and a red, and another part there is green, green and yellow. And then induce the metaphase 2 and go to anaphase 2. The blue one and the red one, blue one, red one, they are separated into two sides of the cell, and they also as the same as the green and yellow. And after the anaphase 2, each one cell has one blue, one red, one blue, one red. And for another cell, one green, one yellow, one green, one yellow. And so I'm gonna talk about some differences between meiosis and mitosis. So first, stages. Mitosis has one cycle, meiosis has two cycles. Cycle basically is prophase, metaphase, anaphase, and telophase. And for meiosis, they are like prophase 1, metaphase 1, anaphase 1, telophase 1, and then go to meiosis 2, anaphase 2, and telophase 2. And for my picture, I didn't conclude every single part of the process because it will be super confused. And the second one is procedures. Mitosis is 2n from too small to make too small n identical, and meiosis is 2n make 4 small n, so they are different. But remember that n and n, whatever in meio or mitosis, identical. Like they're identical. They're identical. Every n are identical. And the third thing will be features. Like there are two things it's only meiosis have. One is synapses. It's pairing homologous. Remember in this stage, um, they are synapse. I will use this color to show. There are something in between. Synapse is basically just pairing homologous or hold homologous together. And second thing will be crossover. I'll use the maybe orange color to show crossover. Crossover is basically the exchange different homologous um, information. For example, there is a line. Uh, I'll use another color. For example, we can use the red and yellow. After in the meiosis one, they will exchange little pieces of them for like going to here and going to here, and the line will become this long, this long. Will be like this. Each chromosome will contain a part of others information, right? So this one is crossover, basically exchange the genetic information. Exchange genetic information. And crossover will not happen in the somatic cells because every somatic cell has its own genetic information. All of them are same. The last one will be usage. Mitosis is for somatic cells. Somatic cells, basically every cell without sperm and egg cell, like without germatic cells or germline cells. And meiosis is sperm and egg cells.